one panelist today, and we are just going to wait maybe a minute for more people to join this talk. And in the meantime, I just want to kind of let you know that this talk, as well as all the talks that we had during this month, you can go back to them and visit our Hawaii Invasive Species YouTube channel. This talk is being live streamed, so you can go back to it later and check it out, or share it with your friends and family. And if you have any questions for the speaker, Heather Spalding, during the talk, please, you can write them in the Q&A box in there, and then she will be going through the questions once when her talk is done. We will also have a quick poll in a little bit that will be launched so that we can learn a little bit more about the demographics, so that we can better do things in the years to come. So I think we are probably a minute in, and maybe it will be time to launch the poll already. Chelsea Arnaud is helping us with the logistics. So if you can take a few seconds and complete the poll, please do so. All right, and I think that is the only one that we have, or I'm not sure if there is another one.
That's the only one, Layla. There's 3 questions on it that people are answering right now.
All right, great. All right, and as I said, like, if you have any questions during the talk, please ask them. I will introduce Heather Spalding. She is a good friend of mine, and she has a bachelor's degree in marine science from Long Island University. She earned her master's degree in marine science at San Francisco State University. After that, he came to Hawaii to earn her PhD at the University of Hawaii at Manoa. And she was a postdoctoral research fellow for a few years here at UH Manoa, for working on several different projects. She is currently an assistant professor at the Department of Biology, College of Charleston, South Carolina, and she's still an affiliate faculty in Ecology, Evolution, and Conservation Biology program here at UH Manoa. And today she is going to talk to us about new invasive algae, and she's going to tell us all about it. So take it away, please, Heather.
Okay, thank you, Layla. Oops. Let me share my screen, and away we go. Okay, thank you all of you for joining me. I think I've raised my hand, but that's okay. I'm gonna leave it up. There you go. So what I'm going to talk to you about today is the limu, or alga, Chondria tumulosa, and how it overgrows reefs. So, but first, I'd like to acknowledge many people who contribute to this research, and this isn't even all of them. The research on this alga has definitely been a group effort, and it's by all of the incredible scientists and resource managers and students that have been able to do research on this alga, that we've been able to find out some of the information that I'm presenting today. And so by working together, we've really been able to at least start to get a better understanding of the different dynamics in the biology of this alga. Okay, so let's orient ourselves. Where exactly am I talking about here in the world? Well, what I'm going to focus on talking about is the Northwestern Hawaiian Islands, which are located within the Papahanaumokuakea Marine National Monument. So here you can see the dotted line, which is the Papahanaumokuakea Marine National Monument. It's one of the largest protected marine areas in the world. And as you can see, it's to the northwest of the 8 main Hawaiian islands, and it has over 130 islands and atolls within its boundaries. The area that I'm going to be talking about primarily today is up there in the yellow dot, and that is Manavai, otherwise known as Pearl and Hermes Atoll. But I'm going to use the Hawaiian name for it in today's talk. So I was lucky enough to be able to go on a research cruise with the National Oceanic and Atmospheric Association to try and monitor and collect the algae or the seaweeds up in Papahanaumokuakea Marine National Monument. And it was a real honor to be able to go to this spectacular location and some of the other islands and atolls. So if we look closer at Manavai, what you can see is that it's a true coral atoll. So you can see the ring of remnant coral around the edge, and then it has a few scattered small islands to the south and then up in the upper west right there. But for the most part, it's a large coral reef. And now this picture is from NASA. And the reason I like it is because what you can see is all of the currents, the currents coming up and out of the lagoonal system, both to the west and then up there to the north. Now be sure to remember that because I'm going to talk a little bit about that more later. In terms of size, it's actually about the size of Oahu. So it's the second largest atoll in Papahanaumokuakea Marine National Monument. And so, for instance, it's the dominant northwest swell, which means that northwest area, you can sort of see the white of the waves breaking in this picture. It really gets hammered in the winter with that large swell. And you also have the wind coming around in that direction, so that the north and the western sides of the islands get a lot of wind, so it's difficult to drive your boat up through there. And so we spend a lot of time in the southern portion of the atoll that's more protected from the wind and waves, and then we are able to go out and take small boats around to the other sides of the island. So in terms of size, if you're used to being on the water in a boat, if you're launching from a large boat from the south side in the protected area, it's going to take you a couple hours to motor all the way and get to the north. And then you could maybe dive that site, maybe another site near there, and then you're going to have to drive back, and that's your whole research day. So it's a fairly large place to work. And what's challenging is if you look in the inside in the lagoonal area and around the edges, it sort of looks like it's made of a winding matrix. And that's because it is like a winding matrix. It's the maze. So you really have to go slow. There's a lot of coral bommies and things that come up. And it's like being in a maze trying to get through it. So you actually can't just cut across the lagoon in order to get to the other side. You really generally have to go from the outside and be very careful. So for me, as a marine biologist, what I think about is what's underwater. Now, Manavai has, Hawaiian monk seal populations and turtle populations, and also really pretty and impressive and gorgeous nesting bird populations, but I like what's underwater. So, and one of the things that's most impressive when you go to Manavai is the fish. And that's because it has an intact food web. So you have large alua, and you have these big schools of herbivorous fish like these manini. So for instance, as you see in this video, there you go. So this is one of the school of fish that you could encounter when you were there. You can see them going and grazing on the bottom. You've got a few little pieces of algae that are upright, few bits of halimita right there. But you can see the school of herbivorous fish. On the striped ones are manini, going through and eating all of the small algae that's growing on the reefs. The pink there is a lot of non-geniculate coral and algae, the encrusting algae holding the reef together. Manavai has areas that have a lot of coral, but for me, as someone who's a phycologist that studies the algae, it also has some really neat algal assemblages that we found both in shallow water and some unique limu that occurs in deeper water in the mesophotic as well. So on the first day in 2019 that I was able to dive at Manavai, it was a dive to about the bottom depth was around 60 feet. And as you were descending, what you could see is to the left of the screen, you see that yellow coral, that's a Parietes lichen. And then there's some scattered macroalgae around there. But as I was descending, what I saw in this little depression where you see me right here was this kind of dirty looking mat of what I knew was a type of red alga, but not one I was really familiar with. So you can see me sitting here taking a picture of that mat in that nook. And the picture, this is the picture that I took when I was diving. And what you can see is there's sort of, like a multi-axis matted looking alga. It was pretty heavily epiphytized, which told me it was likely had been there for a little while, but I didn't really recognize it. I didn't know what it was. So I took some, I put it in my collection bag, and we went up from the dive. On the next dive, we were going to dive in a place which is located in the Northeast. It's called Engine Block because there is literally an engine block on the reef from a boat that crashed there. All that's left is the engine block, but it's a very shallow dive. It's about 1 to 2 meters depth. And it's typically has a high abundance of Montipora coral, like what you see here. The common name is rice coral. And so on this dive, this is what I expected to see, with the occasional clumps of algae scattered amongst the Montipora. But instead, when I flipped over on the boat and put my head in the water, what I saw was this. And it turned out, if you recognize it from the previous dive, it was the same species of red alga from that deeper dive. Only in this case, it looks like the alga has been at this site for a lot longer. It was much more abundant. And so what it formed were these very thick mats that you could actually lift off of the reef. And so in terms of what this site looked like, here you can see these mats growing over what used to be Montipora. And you can see them slightly becoming unattached and sort of moving with the water and the current. What it's doing is it's basically abrading the substrate down below. You can see it in that little puka right there. You can see a little Montipora that's still alive, formed a little hole where the alga grew around it. With that abrasion, I would be surprised if that coral was there today. So you can see a few little fish swimming around in there. Those are damselfish, which tend to like areas of high algal abundance. All right, so at this point, after the second dive, I knew something was wrong and that there was a big problem. Because I had never seen this alga before, and it was growing in exceptionally high abundance. These sites are monitored regularly by the monument and by John Burns' lab and his students and technicians. So they have a really good idea of how these reefs change over the scale of years. And previously, they have no record of these sites looking as, like this with this high percent cover. So if you tear away one of those mats, this is what it looked like underneath. So basically, the coral is dead. Underneath the mat, there are a few patches of live non-geniculate coralline algae or pink crustose coralline algae. But again, that's a group of algae that can grow under very low light conditions, and they're pretty resilient to things like smothering. So that's not surprising. What you can see is that there are little pieces of holdfast that are still attached to the rock, even though the mat of the alga had been removed. And so looking at this a little bit closer, to me, one of the most impressive attributes of this alga, aside from its ability to form these dense mats, is its photoadaptation and its differences in morphology. So at the top of this mat, you can see where it's exposed to high light. And so it's very lighter colored, and it also has it's densely matted. And then down below, you see the pigmentation quickly changes to more of a shade adapted color. And it has elongate axes that sort of reach down and hold onto the rock. Now, this particular mat is getting ready to detach from the substrate. And so you see that big gap right there between the mat and the substrate of, what is basically dead coral. So this was really shocking. So here we are, we discovered this invasive alga. And like I mentioned, luckily, John Burns' lab had photogrammetry from this exact site. And so by going back and looking at it, they were able to ascertain that this unknown red alga actually was present as early as 2015. So here's a 3D photogrammetry image of that exact reef where I just showed you the pictures. And I know it's hard to see unless you zoom in, but basically the areas in yellow is where they found small patches of this alga. Now by 2017, this is the same reef. You can notice there's that small keyhole formation on the right, on the top image. If you look down, you can see that same small keyhole formation on the right and the one below. In 2017, it turned out that now that they knew what they were looking for and they had the signal for it, they knew that we're able to calculate that over 50% of the cover, and by 2017 at this site, was this alga. 2 years later, when we were there in 2019, it was near 100% cover in most of this area. So a lot of dramatic changes in terms of how quickly this alga was able to invade into this environment and overgrow this particular coral reef. In 2017, I didn't circle all of it, but basically everything that looks sort of that orangey yellowy color is the Chondria, or sorry, the unknown alga. So the real question, so we're sitting here, we just found this alga. We have no idea what it is. I don't have a microscope on the boat, so I can't cross-section it and try and look at it to get at least identify it to the family level. So the real question was, what is this? Right? It didn't look like any alga that I had ever seen from the main Hawaiian Islands or anywhere else in the Pacific. I will tell you, it fragmented really easily. So when you held it in your hand and squished it, it broke apart just like spiny weed or Acanthophyr spicifera. But it looked, with all the spinose protuberances all over it, it reminded me kind of Capophycus and Eucheuma. So I was like, maybe it's that, but that's a whole different type of group of algae. So we really didn't know when we were out there what it was or where it came from. Luckily, when we got back, Allison Sherwood's lab and our colleague John Huseman were able to use molecular techniques in order to identify it. And it turned out it was a new species that belonged to the genus Chondria. And so we named it Chondria tumulosa. Tumulosa refers to sort of the mounds that sort of roll across these reefs when a reef gets covered with this alga. So looking a little bit more closely on Chondria tumulosa, it is in the Rhodomelaceae. So it has a filament that runs through the middle of it, surrounded by pericentral cells. And it, in terms of the family, it's in the same family as Acanthophyr spicifera, as spiny weed. So I could see why it sort of is similar to that in some ways. In terms of taste, it's kind of country. I detected a slight peanut aftertaste. Nobody else did. That might have just been me. But it tasted okay. But we really need a lot more information about it in terms of its ecology and physiology. But the catch here is that it's new, which makes it really difficult. And it makes it difficult for a few different reasons. Number one, because it's a new species, we don't know, anything about it. It's mat forming, which is really weird for the genus Chondria. Chondria species typically don't form dense mats. Now, you would think that if it was forming dense mats somewhere else and would have the potential to overgrow a reef, that someone would notice it in the Pacific if it came from somewhere else. So there's a lot of mysteries around this alga. So because it's new species, and we really don't know if it came from somewhere else, or maybe it was a cryptic native alga that just went haywire due to some sort of changing environmental conditions. We can't technically call it invasive. It's cryptogenic. We don't know where it came from. But what we did feel comfortable saying is that it had a lot of invasive-like traits. So right now, that's how we have it classified, as a nuisance or cryptogenic alga with invasive-like traits. All right, so let me talk to you a little bit about what we do know about this alga thus far. So this is a dive that we were doing, and the bottom depth was about 80 feet, a little bit more than 80 feet. So based upon what we knew from its distribution at engine block down to surveys that we did while we were there at Monavai in 2019, and then later surveys that we came back and did in 2021, we know that it can occur to at least 21 feet. But I say at least 21 feet because this is that deeper dive that I'm talking about. And if you'll notice, it may be hard to see, but all of the brown, lumpy looking stuff in this picture is chondria. So you're looking at nearly 95% cover chondria on this spur and groove reef. So it continues all the way down to the sand. So it requires hard substrate to grow. We did not find it growing in sand. And so typically, if you look at the bottom depth limit of an alga, it'll start to get less abundant and then slowly decrease in abundance till it reaches a certain limit, which is typically the lower physiological limit for it to grow in terms of light. But it does not appear to be limited in abundance in this particular spur and groove reef. So I think we really need a little bit more information about what its lower depth limit is. Another aspect about it growing in these spur and groove reefs is that we noticed that there was a lot of fragmentation. And in these spur and groove reef areas, there were large currents that come through these spur and grooves. And oftentimes at the bottom of these reefs, you'd see fragments like the one floating up in the middle of the screen right now that broken off of the chondria and were rolling around. And so there appeared to be ample opportunity for transport in these communities via these spur and groove reefs and these strong currents that we often felt. I have to admit, it's really interesting diving at Monavai because you could be on a dive and facing one direction and you're feeling okay and kind of warm, but it's a regular dive. And then the water will start to shimmer and you'll feel this cold wave underwater wash over you in the opposite direction, and your whole body turns while you're diving to move with the water. And now you're facing the opposite direction and you're freezing because the water is so cold. There's a lot of interesting oceanographic currents that are influencing Monavai. And we really can't help but wonder if maybe these conditions, bringing in nutrient-rich water, possibly from deeper water or across the atoll, could be influencing the success of the chondria, especially in these spur and groove areas. So Andrea Kealoha at UH Maui is looking at the oceanographic conditions around Monavai, and so I'm hoping she can help us better understand what's going on here. So because of these fragment issues that I was talking about, we quickly learned that when you dive with chondria, you have to be very careful. These fragments come apart, they travel in the water column, they get caught in your hair, they get caught in your goodie bags. And so what we developed was a procedure and how to handle chondria mats and pieces of chondria. And so one thing is that they have to be double bagged. So you have to take a mat, put it in a bag, and then put that in another bag when you're diving. This is Taylor Williams right here handling the infamous mat, our one big mat piece that we took back to the main Hawaiian Islands after it was frozen so that it would be dead. So bagging it, when it comes into the boat, it has to go into its own special cooler so it doesn't break apart or get into the boat. And then when you're in the lab, it needs to go straight into the lab so it doesn't get onto the deck of the boat or get blown overboard. So you have to be really careful when you're working with this particular alga. And so based upon our experiences, we were able to provide advisement on the development of BMPs on how to handle working with this alga. And like I said, though, the fragments were everywhere. So when we first discovered it, and we at engine block, and we removed some of the mats to try and get a better understanding of what it was and how it was attached, we really did get fragments in our hair and in our gear. And even though we tried to wash off before we got into the boat, we noticed that again, it's very easy for these fragments to get caught in your dive gear. And so at this point, we're on the boat, we're at Montevideo, you know, we're supposed to continue on this research cruise all the way up to Midway and Curie, and then all the way back down the atoll. And what we realized is that we can't, you know, the cruise has to stop. You know, we just discovered a highly invasive alga invasive leg alga overgrowing the reef. We don't know what it is, and there's fragments of it everywhere. And we really have to figure out some really important questions like, well, how are we going to kill it? And it turned out that, you know, when we move between atolls when we're diving, they have a very good procedure where you have to soak all of your gear in a percentage of bleach. But what we found is that when we soaked our gear in the bleach, the alga, the Chondria didn't care. It looked perfectly fine and healthy. And it was like, yeah, bleach, doesn't bother me. So what we found is that we really needed to change things up. And we needed to figure out how are we going to kill these fragments that we have that are being created and working with this alga that are on the boat and in our gear. And you know what, and also we're trying to sample it, you know, that in our lab areas where we need to try and collect samples of it. So in collaboration with Brian Hawk, we were able to do trials basically. And again, I kid you not, this alga just gets everywhere. So with Brian, what we did is we were able to create these experiments where we took small glass vials and took fragments of the alga. And then each bottle did different trials, replicated N equals 10 per treatment of different concentrations of bleach, different durations of time, different size fragments. And after lots and lots and lots of fragment trials, what we found is that over time, you know, 6% bleach, which is double what we were typically soaking our equipment in, works a lot better. But it takes at least 10 minutes of 6% bleach for it to actually kill the Chondria fragment. So if you look on the left, you can see one of our trials with our fragments. Each row is a different alga, or is an alga that was soaked for a different duration of time in 6% bleach. So you have the 1 minute, the 3 minute, the 5 minute, the 10 minute, the 12 minute. And you can see at 10 minutes, the algae finally turns white, which in my mind represented death. I was happy with that. So we did all of these trials and found different ways to be able to kill the alga. And to be honest, more trials need to happen. Unfortunately, like I personally do not want to leave my dive gear in 6% bleach for 10 minutes because it's going to eat up my gear and degrade the equipment. And so we really need to continue working on this and come up with different types of recommendations, like maybe 3% bleach for a half hour or an hour. Even that, you kind of worry about it hurting your gear, especially if you use it to breathe on underwater. So we really need to develop more BMPs to figure out how best to handle this alga, since it appears to be fairly resilient to bleach. Okay, so just to give you an idea of how this reef looks when it invades, and this is something that we came to appreciate in doing swimming surveys on the reef. So this is another type of Porites, and you can see it abundant here, and there's a few fish here and there. But basically, you would start to swim, and you'd swim through this coral-dominated area. And what you would start to see is that in between the coral heads and in between the flutes of coral in this case, the chondria would establish and then just start to fill up and grow up through the coral until finally you can see just the tips of the coral surviving. And then finally, the chondria would overgrow the coral. So if you look over on the left, though, you'll see a little hole, and I'm going to talk about that in a moment. So in this type of reef that's been basically overgrown with the chondria, what we noticed and what it does is that it really fills in the pukas and takes away the 3D complexity of the reef and creates these rolling, soft sort of hills of Chondria tumulosa that are very different structurally than the original reef, which tends to have a lot more rugosity, an area we presume for fish habitat and other organisms. Now, when you find these areas that are heavily overgrown, they do have small little bits of algae at the top that poke up, that survive, but there's not much. So just to give you an idea of what one of these reefs look like. This is at about 30 feet. And you can see, as you're swimming along, you can see how it sort of overgrows the reef. You'll notice there are a few fish, but those are damselfish. You can see one small patch of Parietes lichen on the right-hand side that's yellow that hasn't been overgrown yet. And again, the fish that you're seeing in here are all damselfish. So these are fish that are actually using the algae for habitat. So the Chondria, we observed it overgrowing every species of coral that we encountered at Monavai. So here it is filling in between and overgrowing pieces of Parietes lichen. Monavai has a high abundance of native microdictyon beds, which is this green net-like alga. And sometimes we would find a bed of microdictyon and we would get excited because we're like, oh, there's no Chondria here yet. This hasn't been invaded yet. And then we would collect some microdictyon samples for different studies, bring them up. And then when we were pulling through them in the lab, we would find pieces of Chondria. So just because you don't see it doesn't mean it's not there. So in this case, you can see how the Chondria will just overgrow whatever native algae are present, and it can form these draperies over the sides of rocks or corals. And so basically creating sort of like walls of Chondria down the side. So here's an example of that, where this was a fairly steep wall with small caves underneath. And what has happened is the Chondria has just overgrown that. Now, if you look to the right, you can see again one of these damselfish. The only fish that I saw actually using the Chondria for habitat. The other thing is that what was strange about having a large invasive like algal outbreak at Monavai is that remember the pictures of all the manini that I showed you earlier. This is an intact food system. There shouldn't be an overabundance of a particular alga in this system unless the alga is not palatable or the fish don't recognize it as a food source. So I feel like this is pretty well illustrated. This is in one of the spur and groove areas. This is a large school of kala, of nazos. And they were heading over for this spur and groove, which was over across from us. And then they turned, which was very convenient. There's a kala or alua, sorry, right there. So then they turned and came back towards us. And as they did, they came over this reef that we were on, which was covered in Chondria tumulosa. And at first, what you can see is they just swim right over it. And then towards the top of it, it does. At first, we're like, oh, look, they're grazing on the Chondria. But then what we realized is they're little pukas where the Chondria wasn't growing. You can see them more now right here. You can sort of see where it's sort of sedimenting. And what we realized when we got closer is that the fish were actually targeting those pukas and swimming right over where the Chondria was located. And so as of right now, the only organism that we've seen that has actively grazed on the Chondria has been one large sea hare. I found it in a hole and it was sort of projecting out and eating out of the hole. It had gotten so fat, I think it was stuck in the hole in the reef from eating all of the Chondria. Other than that, I haven't seen any fish actively graze upon it. All right, which brings us to, its distribution. So in 2019, we surveyed several areas around the atoll, and these are shallow areas surveyed that you see here in black. And what we found is that it's basically located along every north, south, west, east part of the atoll. In 2021, we went and surveyed more of the southern areas, and we found it there as well. Now, the good, at that point, it was absent within the interior of the lagoon. But when we went back in 2021, we found that this had changed. Now, on the plus side, at mesophotic depths, which we typically define as, greater than 40 meters, 30 to 40 meters. Now, we did not find any chondria at those depths. So at least at the sites that we had surveyed. So that's encouraging that at this point of time, they don't occur that deep down into the water. But again, we do know they occur down to at least 21 meter depths. Looking at the delta N15, which is a stable isotope collected from the algal tissue, what we found is that they were what we would consider sort of normal open oceanic levels of delta N15 at these 6 different sites that were surveyed. But when we look at the percent nitrogen within the chondria material collected from these sites, what we found is that up around site 4, it was on average over 2% nitrogen, which typically means that it was enriched in some way. And we went back in 2021 and sampled additional sites. Unfortunately, site 4 and 5 were the sites that we couldn't get to because of bad weather. So I'd really like to go back and see if this pattern continues to persist. But remember that picture that we have from NASA, those currents that were coming out from the north were coming out around site 4. So in the interlagoon area I was talking about, this is this high density of chondria that we found floating in the drift. And so you can see some of the divers are snorkeling up at the top to record it. So this, we call it the Chondria Highway because it was this large band of drift chondria that had rolled up off of the reef. And so this was located in the eastern portion of the lagoon. Additional research by Keo Lopes and Tomoaki Miura found that this is actually visible from satellite imagery. So after our cruise in 2021, unfortunately, chondria was also found at Kauai or Midway Atoll. So now we know at this point in time, it's found at least 2 locations within Papahanaumokuakea Marine National Monument. So I'm almost ready to wrap up, but I just want to say that this work is just beginning. So I'd like to recognize Taylor Williams and my colleague Stacy Cougar Hadfield at University of Alabama, Birmingham. They're working on the population connectivity and reproductive state of the chondria. And they're going to try and answer whether or not this alga is entirely clonal, like is it one piece that drifted in and now that's overtaken the entire atoll, or is it sexually reproducing, which we can look at looking at ploidy. And more important, and as importantly, is what we have now at Kauai, is it the same population as what we have at Manabai? Connectivity is important to understand. We have boats and vessels and equipment moving between these different islands and atolls, and is that safe in terms of the algal propagules that may be in the water attached to these vessels or equipment, or even fragments? So do we manage them as one entity, or do we manage them as 2 separate entities? These are important management questions. The other person I'd like to acknowledge is Gabby Kuba and my colleague, Dr. Heather Fullerton. They're actually working on the microbiome, the bacterial microbiome found within algae in the main Hawaiian Islands, as well as in Papahanaumokuakea Marine National Monument. And their work has been looking at the bacterial microbiome of chondria as compared to other native algae in shallow water, as well as mesophotic samples. And so that paper right now is in preparation. And so finally, to wrap up, what do we know now about Chondria tumulosa? Well, what we can say for sure is it's a new cryptogenic alga, but it definitely has some invasive-like traits. And I hope by showing you some of this video today that you'll understand that. We know that it forms thick mats. We've measured mats up to 21 centimeters in depth. I'm not just talking about the lower depth limit in meters, but I'm talking about how thick it is in terms of the mat. And it does smother native corals as well as the native algae that are a critical food source for herbivores in this ecosystem. And now we know it's widespread around Manawai from 1 to 21 meter depths, and now it's found on Kauai as well. All right, so I would really like to appreciate all of the support. I really appreciate all the support that we get from our different funders for this. This is NOAA and the National Fish and Wildlife Foundation, as well as NSF. Our partners with NOAA and U.S. Fish and Wildlife Service have been incredibly helpful in helping us to continue this work. And of course, a very special thank you to Papahanaumokuakea Marine National Monument for giving us access to this incredible place. All right, do we have any questions? I'm assuming Layla, should I stop the share on my screen?
Yeah, it's up to you. Like maybe you can stop sharing so that you can answer some questions. Thank you so much for the great presentation. And we have Taylor Williams here as well. You can turn on your video as well. She is she's a graduate student with Heather Spalding, and she is studying this invasive or this alga, and she did her bachelor's degree at the University of Hawaii, so she will be helping answer any questions you might have. And maybe to start things off, are there any other Chondria species here in Hawaii, and are they all invasive or?
Yeah, no, it's a great question, Chondria, about Chondria. So Chondria is a genus where we have several native species in Hawaii, both in Papahanaumokuakea Marine National Monument, as well as in the main Hawaiian Islands. But to be honest, they're typically not in high abundance, and they're definitely not mat forming. They can sometimes be cryptic, so they're not always obvious, but they're there and a present part, typically part of the turf community, which is why it was very strange for us to see Chondria tumulosa growing in its morphology and as abundant as what it was. And that's part of the trick with this alga, is that it is very easy when it's in low abundance to confuse with native algae. So it's just when it gets really abundant, at which point it's almost, I don't want to be catastrophize it, but it almost feels like once it starts forming mass, it almost feels like it's too late. That means it's invaded, it's been able to disperse by fragments or by small pieces of mat. At that point, it's probably a lot more abundant than what you think it is.
And Taylor, what is your work focused on?
Thanks, Layla. Yeah, so I'm looking at the reproductive system of Chondria tumulosa. I'm really interested in like how the reproductive system affects the genetic diversity throughout the known chondria populations. So trying to decide if it's, or trying to determine if it's sexually reproducing, if it's asexually reproducing, and trying to see how exactly that is affecting the population genetics, both at Mauna Vai and at Koihalani.
Great, and we have one question. Is there work being done at the moment to remove this invasive alga?
Taylor, would you like to answer that?
There is no current work being done to remove this species. I think that there's been a lot of talk about different strategies for removal, but none of them have seemed all that practical yet because of the location of this invasion or this alga. It would be really challenging to actually create a management strategy or a removal strategy because it's a 5-day ship transit from Oahu. Just removing it and then first just getting access to it is a challenge, and then figuring out how to remove such large mats can be challenging. And then what do you do with such biomass once you have it back on the ship are all questions that we're still trying to figure out right now. So no, there's been no work to remove it quite yet.
And we have another question. Have you done any further analysis of the photosynthetic properties of Chondria?
Yeah, so thanks, Christian, for asking the question. I personally have not, but luckily in Hawaii, we have a fantastic physiologist, Dr. Celia Smith, and she was able to come out on our last cruise and collect some samples. So I'm hoping that some of these pigment analyses will be forthcoming, and so that we can better understand some of its strategies. Luckily, there Dr. Smith's lab has done some really neat work with photoadaptation and other invasive red algal canopies like Gracilaria salicornia, which also forms dense mats and is an invasive red alga in Hawaii. So we don't have that data yet, but I'm really hoping we can get it.
Great. There is another question. What reproductive cycle has been discovered in Chondria?
Taylor?
So what we know so far, all rhodophytes, so all red algae have haploid diploid life cycles. And so at some point, well, so humans have diploid life cycles, meaning that we have 2 copies of our alleles. There's this long-lived diploid phase, which is what we are. And then there's like the reduced haploid zygote, right? That's the diploid phase. Red algae have haploid diploid phases. So they can have this long-lived haploid individual and a long-lived diploid individual, which has a lot of impacts on the genetic diversity, which is part of what I'm studying. And so what we know is that it is asexually propagating via fragmentation. And we know this because we've observed it both in other red algal species and in Chondria. We have also found tetraspores. And so there is some meiosis occurring, some indication of some sexual reproduction. We are still working on that data, hopefully forthcoming soon. But that's what we know right now, is there is some tetraspores, some indication of sexual reproduction, but mostly what we're seeing is asexual fragmentation. And based on some limited genotype data, we do know that it's not all clonal. So there are separate genotypes indicating, again, sexual reproduction is occurring. We just don't know at what level yet.
Great. Thank you so much for answering the questions. We'll just wait a few more seconds just to see if there is any other questions popping up in here. And there is. Are people able to donate to research like this in Hawaii?
That would be a great idea. I don't know. I should know exactly how someone could do that, because it would be really helpful. I mean, right now, where our funding comes to be able to study this alga is we get support from the National Fish and Wildlife Foundation, and they provide grants, which we then apply for competitively to try and get the funding to be able to dive and go up there and use boats and do the analyses. But really, access is truly granted through the support through boat support and plane support through organizations like the NOAA Papahanaumokuakea Marine National Monument, as well as the U.S. Fish and Wildlife Service. And so it may sound funny, but for us right now, the best way for us to be able to get up there is to make donations to those organizations so that they can support the research that's needed to manage the monument. But I will look into that to see how you would donate to support this work. But I think it's important to work. The thing that's hard to wrap your mind around is that I could see how someone would look at these videos and be like, bah, it's up in the middle of the monument. You know, that's, you know, hundreds of miles away from where we may be in the main Hawaiian Islands. But given what we saw about how this alga floats, and given what we saw about how it fragments, and given that we've seen how quickly it can overgrow a reef, we really worry that it's only a matter of time before this will hop its way down through the Northwestern Hawaiian Islands and end up in the main Hawaiian Islands. And given that how this alga is behaving in a system with an intact system of herbivores, how is this going to react when it hits a location like the main Hawaiian Islands that are more heavily eutrophied and have high concentrations of humans living on land and changing the reef in ways, not just in terms of nutrient regimes, but also in terms of overfishing. I mean, the only place that I've seen schools of fish like that have been at either like when I'm diving at 100 meters, which I don't recommend for most people, but we still have some large schools of fish down there in the main Hawaiian Islands or in Hanauma Bay. So it's, you know, it's a real danger. And so I think that the more that we can learn about it now while it's isolated, I think it will help for management purposes when we, you know, when and if it does reach its way to the main Hawaiian Islands. And so like even just doing presentations like that, where we can help people to start get a visual image of what this looks like. Hawaii is lucky to have so many people that are in the water and aware and dive and snorkel and are, so knowledgeable about limu, that I think that the more people that can learn about this and understand that, hey, it's there and this is how it grows and watch out for it, I think that's just as valuable as anything else.
Great. We have one other question, two more. How long did it take for Chondria to go unnoticed and invade in the Northern Hawaiian Islands?
Taylor, do you want to talk about timeline?
Sure. So in 2015 is the first record that we have of Chondria tumulosa, pre-identifying it as Chondria, of course. We first have it on 3D photogrammetry in 2015. Is that right? We have a sample from 2015 and 3D photogrammetry from 2015. And since then, we also have 3D photogrammetry from 2017, 2019. And then we didn't get 3D photogrammetry in 2021, but hopefully we'll have a follow-up in a couple years to see the progress. But we first found it in 2015. We just didn't find it in as high of abundance until about 2019. So it's very limited in 2015.
Yeah. I will say in 2016, luckily, someone from NOAA, Louise Giuseppe, was on a cruise and she collected algae and noticed it and noticed, unknown red alga, and took a sample and froze it. And so it was her samples from 2016 that we were able to confirm that, it was definitely there in 2016 and, in the same places where these maps were. So, using that information, 2015 is the earliest record that we have of it, but it was likely there a little bit before that, but just not in high abundance.
All right, we have another question. How often are Manawai and Kauhuhelani monitored?
Yeah, I'll take that one. That's a great question, and it's a tricky one because the Papahanaumokuakea Marine National Monument is in charge of monitoring and managing this huge area. And yet right now, they don't even have a research vessel. They don't have their own boat to be able to go up and monitor, an outbreak of a highly invasive alga of unknown origin. So, we just had Hurricane Walaka that leveled Lalo. We've got bleaching events that are happening, and then to try and manage this whole area without boats, that's the real limitation. And from my mind as a scientist who wants to go up and help and continue to study this system, they desperately need a boat. And so a research vessel where scientists can help and monitor and maybe attempt to even manage at some level what's up there. So right now, like Taylor was saying, you try and go up once or twice a year, but right now, like we don't have access this year, despite best efforts of everyone involved who's interested. It's just really hard to get a research vessel in the Hawaiian Islands to be able to get this research regularly. So donate to NOAA so that the monument can get a research vessel so we can go up there and do research and figure out some of this. Part of the reason I was on the initial cruise where we discovered this alga was to document limu diversity. And on that cruise, before we encountered the Chondria and got shut down and basically told you can't go anywhere until you figured out how to kill it. We discovered several new species of limu. Like we were doing mesophotic. I was not personally doing the mesophotic dives, but monument staff were doing mesophotic dives, and we were discovering new limu left and right. And we even found some new limu in shallow water, another new species of red alga. It's an incredibly diverse area that we're still at the tip of the iceberg of understanding the level of endemicity and new species in this region. And so that's part of the heartbreak for me, is that to see something that's just overgrowing the reefs while we're still documenting what's there, it's difficult to see underwater in a place that, to be honest, is magnificent in the areas where it's not growing.
Great. I think these are all the questions we had. I think we had a really great questions. And thank you so much again for your time. And yeah, you are just getting thank yous in the chat. And yeah, thank you so much for everything you do. And hopefully you continue to do the work you are doing, because it's very important. And with that, I would like to thank everybody that tuned in. And yeah, thank you again. And we'll see you sometime, hopefully soon, and keep up the good work.
All right.